Okay, good morning. And yesterday we have learned about homologous recombination, the mechanisms of how DNA exchanges in terms of strands. Okay, and I am proud of you that you all have done the assignment well, and I'm sure now you understand the concept of it. So in today's class, we will quickly go through the we will review what we have learned in the last class, and then we will see some examples. Those examples will be exciting, of which we will learn about how yeast can change its sex type. Okay, we will learn about it. So to start, the double strand break model is the most accepted model in the current day, because it has an explanation of how double-strand DNA breaks can be made. It could be an accident, it could be induced chemicals, or it could be a way of by some proteins. We will see them. So the first step is to have a double-strand break. Second is processing to generate 3' overhangs. And then we have strand invasion, where the 3' overhang makes a strand invasion like this. And there is a displacement of the strand from the bottom DNA molecule. And then we have two replication forks arranged. This is one replication fork, you can think of it that way. And this is another one. You have a primer template junction, which can be taken up by DNA polymerases, and polymerization will occur. And during that process, the holiday junctions will move further apart like this, right? That is called as branch migration. Sorry. So the first is alignment, double strand break, processing, strand invasion, and then you have, in double strand break, we also talk about replication fork formation, replication, initiation maybe, or sorry, I should not use initiation. So here replication will take up polymerization to fill in the gaps, and then you have branch migration of the two holiday junctions, and then we have resolution. We have learned about how to resolve it. It can be done on site 1, where you're cutting, if you're cutting these two strands, then it is site 1. If you're cutting the internal ones, that is called as site 2. We have also learned what is a non-crossover product, or splice, or patch product. We have also learned about what are crossover products or splice products. I hope you have done the assignments, and some of you people haven't described what kind of products are formed. As opposed to two holiday junctions, here we only have two holiday junctions. I gave you as assignment with 3 holiday junctions, and then the consequences could be different. So you need to, just for sake of you understanding, I've given it, and I'm sure most of you have understood the concept. So today we are going to see the biological relevance, okay, or what is the importance of this homologous recombination in biology. We are not going to deal with all the things that are that we know of in biology, but we will focus on some aspects or examples that are well understood. And those similar trends are what you will see in everywhere else, in other forms of life. So to start with, I will take the simplest organism. So here, say for example, there is E. coli, and it will take up nutrition. It has its own genome, okay, and it takes up nutrition and so on. Say there is no more protein or no more glucose and so on, but what there is outside is DNA. Okay, every time when we talk about DNA, we should be wary, or we should pay attention to two things. As I said in our first lessons, DNA is the only molecule, nucleic acids are the only molecule which is matter with inheritable information, right? That is, the information is present as sequence, and that sequence of bases, and that is genetic information. So when we talk about DNA, it is a molecule with genetic information. So, but it is also, it is two things, three things, if you want to talk about it. It is certainly matter. It is made up of matter, atoms, molecules, and so on. It also has energy, because all the chemical bonds have a lot of energy. So it is also a form of energy. Majority of the things that we know of, these two are of course related. The one thing that is additional in DNA is that it is information, genetic information. So if these DNA molecules enter into the cell, what can they be used for? One thing is they can be used to derive energy and to fuel the metabolic processes of the bacterium. And sometimes the material will also be used up to, say, nitrogen atoms and phosphates and other things. They can be used up to make amino acids and so on. There is also one risk, or it can be a boon or a bane, getting the information from outside. Say, for example, if more and more information from the outside is taken up and randomly put into the cell, what would happen to it? It is causing mutations. Always, I forgot to mention when we are talking about in our mutability chapter, is mutations are not always bad, right? Mutations can be beneficial or they can be lethal, or they may not have any kind of meaning at all. When we read about when we read about translation, we will talk about frame shift mutations and other things. I didn't talk about many of them. Some of them, it's mostly about description. We haven't spoken about it much, but we will discuss when we are talking about translation. So keep that open in your minds. So they can be beneficial to the cell, they can be lethal to the cell, they may not have no effect, they may not have any effect at all. But often, most of the mutations are bad for the cell. Okay? And very rarely the next could be they may not have any effect, and very rarely they are beneficial. Okay? So if a bacterium like E. coli is taking up a lot of DNA from the outside and putting it or integrating it, say for example, then it is losing its identity. It might die, first of all. And if it survives and if there is an added phenotype benefit, then it will have enhanced fitness. Fitness is ability to survive and reproduce. And it is relative to other individuals around. So there should be two things are what organisms it's kind of uncertain accomplish, is to remain same and still excel. Remain same in the sense we need to maintain the identity. Okay, we will talk about those things later, but I hope you understood that. If there is, we also learned about source response, right? What is source response? It is no matter what, I still somehow want to survive. I don't want to die kind of attitude, right? In that sense, what we are doing is, but source response, it allows survival probably, but there are a lot of mutations. If there are mutations, it might be very injured kind of thing or slow growing. How can it become normal again? The only way it can become normal is to take up DNA from outside, DNA from its siblings. So now think of the scenario like this. Assume that all this DNA that is here is coming from its clone mates, that is dead E. coli. Say other dead E. coli cells are there, they have released their DNA. Is there an advantage of taking them up? There is an advantage, because through homologous recombination, because source response induced mutations are there, right? All those mutations can now be repaired using homologous recombination. But then how does E. coli know that this DNA is coming from its siblings? And how can it figure out if it is not from somewhere else, which might actually result in more of damage? I hope you realize that if homologous recombination is happening, and if the DNA is coming from other E. coli cells, what is likely to happen? It is likely to be more beneficial. Why is it beneficial? Because it can repair its damaged genes, mutated genes. Okay? Let's see a pathway, and that is called as REC-BCD pathway, REC for recombination. Excuse me. So there are several genes involved in it, and that is what we are going to see about how E. coli can take up this DNA and integrate that information into itself, and thereby allowing its own genome to be, say, recover from the mutations. So there are several steps in it. First step is, say, we learned about aligning, and that is something like pairing homologous DNAs. That is equivalent to aligning when we learned about the holiday model. And then strand invasion. And in some cases, we will also talk about introduction of double strand breaks and processing of DNA breaks, that is to generate 3' OH, and that is where strand invasion happens, and strand exchange proteins. So in double strand model or holiday model, we spoke about all these steps, but we never said how does that happen. And they're all done by several proteins, and those proteins are what we are going to learn and try to understand a little bit about structural basis of how DNA, how the molecular level homologous recombination works. Okay, so remember, in eukaryotes also we have similar ones. I will speak a little bit about it, but not too deep. For concept's sake, you should know about REC-BCD. It actually starts with the REC-A protein. Then there is REC-B, REC-C, and REC-D. After that, we also have REC-UV, AB complex, and RUVC. These are involved in resolution, in branch migration, and RUVC in resolution. Okay. So first, before going into it, we should be able to, in the previous slides, I spoke about how can DNA know, how can E. coli figure out if that DNA is self or non-self. Say these are DNA molecules, and this is DNA from a different type of cell, not E. coli. How can DNA be distinguished by E. coli? Which one is coming from its own type and which one is coming from a foreign type? And that is where these sites are called. These are called as chi sites. Okay, this is the symbol chi sites. It is for crossover hotspot instigator. That is chi. Crossover hotspot instigator. That's one phrase, but the abbreviation comes from there. It is a small sequence, something like this. That sequence in E. coli is called as chi sequence or crossover hotspot instigator. So in all of the genomes, you have, in all of E. coli genomes, you have this sequence in several locations. And there are proteins that recognize them, or we will see it in a later stage. what you will see is that if this is a DNA and it has chi site, if there is a DNA and it has a chi site, and there is a DNA molecule which does not have a chi site, what does that mean? That means this is likely to come from E. coli, and the green one, which are without a chi site, is unlikely to come from an E. coli, not E. coli. Okay, it's not absoluteness, it is probably. This is not a big sequence, so it is, if you take human DNA, you will find this sequence at least once or twice and so on. But the frequency of chi sites matters. In E. coli genome, you will find that these chi sites are well distributed throughout the genome. And what happens is, from here on, say until the chi site, this place, the recombination does not occur. Whereas once a chi site is recognized, from there on, the recombination frequency is higher. You will understand when we get to the REC-BCD pathway. But what I want you to understand at this stage is that you should know that it is crossover hotspot instigator. It's a sequence of DNA that is well distributed within the genome of E. coli. It is useful for distinguishing between which DNA is coming from E. coli and which is not. So here is the REC-BCD pathway. Also remember that the chi site, right? So this is the DNA that has just entered into the cell, and we are going to see how the DNA is going to be processed. So first thing is, any DNA that comes into the cell, it is likely that it should be degraded. Otherwise, if it is integrated into the genome, it might cause mutations, and often the probability of a mutation causing lethality is higher, okay, or loss of fitness. So REC-BCD, this is REC-B, the red one is REC-C, and the green one is REC-D. So these three form a kind of complex, and they bind to the end of the DNA. This DNA is foreign, it has come from outside into the cell. And then the REC-B and REC-D are kind of exonucleases, okay? And they keep on degrading the DNA from the ends. And they keep doing it, and here is the chi site. Chi site is just a small sequence of DNA. And when they come to the chi site, then something is changing. Okay, you also have to understand here that REC-D is a fast enzyme, exonuclease activity, where it moves faster actually on the D. Whereas REC-B is a little bit slower. So when it moves faster, and REC-B, when it encounters the chi site, it loses its exonuclease activity. And so what happens is, what's happening is the top strand is kind of slowly degrading, and when the chi site on the lower strand reaches to the REC-B, right? That is, sorry, REC-C, then the rest of the DNA is being slowly degraded on the top strand, right? Whereas the lower strand is not degraded. Once a chi site is encountered by REC-C, the degradation of that strand stops. What would have happened if there is no chi site here? This strand will also be degraded. I hope now you appreciate how chi site is used to distinguish what from the DNA that is of self and non-self. Self means it's similar, okay? So whether the incoming DNA is homologous or not, I hope we have used the other word. We are talking about homologous recombination, so we need to also use that word. Homology. What is homology? Homology is having highly similar sequences, not necessarily identical, okay? So quickly, rec D, rec B, and rec C. There are several. This is a complex which starts with exonuclease activity and starts degrading. Rec B is faster compared to that of rec C, so there is a small loop-like thing structure also forming here. But once a chi-site is encountered, rec B loses its exonuclease activity, or the degradation of the lower strand is inhibited. If there is no chi-site, the lower strand will also be degraded. Okay? And that single strand is used for strand invasion. Until now, we have seen only the processing of DNA, right? So we don't necessarily have alignment in bacteria. Aligning means in the first steps, somewhere we learned that the two homologous DNA molecules should be aligned like this. That is possible if the cell was diploid. Here, E. coli is haploid in the sense it only has one copy of the genome. Only the other should come from outside the cell. Okay? So it is processed and then something else happens, which we will see now. Strand exchange or strand invasion is mediated by REC-A protein. I hope you also remember where we have learned about REC-A in SOS response. We have learned that it has high affinity for single-stranded DNA. Right? It binds to the single-stranded DNA. We also learned that it has a protease activity that degrades LEX-A protein. So to see the properties of what, or the capacities of what REC-A can do, we will just have a look at this. Say if you have given a double-stranded DNA, linear double-stranded DNA, and a circular single-stranded DNA, and give REC-A to it, REC-A can move one strand from this to this one and result in products, double-stranded circular DNA with a break here and a single-stranded DNA like this. What you should remember is the molecules that we have taken here should be homologous, right? The sequence similarity should be there. If there is no sequence similarity, then they cannot change. They cannot be exchanged. Similarly, REC-A can also do a lot of things like putting single-strand DNA into this one. The reverse of what we are seeing in this case in A. In C, you see that it can exchange DNA from two different molecules. So it's an amazing protein how it makes these things happen, right? What is REC-A? It can exchange single-strand DNA from one molecule to another molecule. It needs for exchange to be mediated. It needs homology. Homology is high sequence similarity. Here is how REC-A can work. REC-A binds to single-stranded DNA, and this process is something called as nucleation. The first contact between the DNA and one of the subunits of REC-A. And then REC-A makes a filament. So 5 prime, this is 5 prime, and here is 3 prime. So onto this side, it adds very slowly, but on the lower side, it adds much rapidly, like, and resulting in a filament like this. It's a REC-A filament formation on single-strand DNA, and it is forming on the 3 prime ends, right? What does that say? Anytime when we process during homologous recombination, processing is to achieve a 3 prime overhang. And why do we need a 3 prime overhang? 3 prime overhang is the one that actually makes the strand invasion. So that is also because of REC-A's structural or the organization of a REC-A filament. Once a filament is formed like this, and it forms more so on the 3 prime side, and that will be used for strand exchange. The next slide, we'll see how it does. So REC-A filament is forming like a coil, right? As we have seen in the previous slide. I hope you keep this in mind, that it is a coiled around the single strand. And then, so if you see the cross section of the coil, you are seeing this is one DNA. Okay, a DNA molecule is in the facing towards us in the cross section wise, and it can bind to a double-stranded DNA like this. And this double-stranded DNA, as if you can see it clearly, if you can see it properly, they are base paired. You see these red dots. This is a double-strand DNA, and this is the single-strand DNA onto which the Recce filament has formed. And now what it does is it moves this to, or sorry, this one to base pair with this one. How can base pairing happen? It will only happen if these two strands were homologous or complementary, then only then there will be change. So the DNA molecule here will start base pairing here to the DNA, single strand DNA that it originally was here, and that is how strand exchange can be mediated. So with the filament, so eventually what is happening, if you see it, what's happening is this, that the DNA molecule, this is a double strand DNA, and here is a Recce filament on a single stranded DNA. And eventually what will happen is you will have a heteroduplex because there is an exchange of DNA. They are homologous in the sense they don't have to be identical, but there should be a large number of sequence similarity. Okay, that is how Recce can exchange the DNA strands between DNA molecules. The next step, this step, Recce mediates the strand invasion, right? And now we have to see about branch migration, and it is mediated by a complex called as RUVAB. It is a tetramer. It is a tetramer of RUA and a hexamer of RUVB. RUVB and two hexamers. RUVB forms a ring-like structures on this DNA, like this, on opposite DNA molecules. And the RUVA is the one that recognizes the holiday junction. Okay, RUVA recognizes holiday junction. I hope you remember that we have drawn holiday junction something like this, right? But if you rearrange it, you can also put it this way. I hope you have seen the video, and then that is good for our understanding. So when you find a complex a DNA structure like this, that is holiday junction, and that is recognized by RUVA, and then it recruits RUVB. RUVB is the motor machine. You can think of it that way. So on one side, it will keep twisting DNA in this direction. On the other side, it will do the reverse of it. So you have a coupling effect, right? One is trying to unwind in one place, and on the other side, you are having one is rotating to wind the DNA. So what happens is more and more DNA will go into this and this one, and more DNA will be taken up to this. So what happens as a result, the branch or the holiday junction will move towards this side like this. So RUVA is a hexameric ATPase. It requires energy to rotate the DNA. It's an active process. Okay, there are ring structure like motor proteins, RUVB, that mediates branch migration. The next step is RUVC, which is the nuclease. I think we remember that we were talking about, when we talk about resolution, right? We talk about two steps. We call this as site 1. I'll just put 1. And when we are cleaving these strands here, we call it site 1. If we are cleaving it here and here, we call that site 2. RUVC is the one that cleaves. Right now, it is a dimer, okay? You have two subunits of RUVC, and they might bind here like this, and they make a cut here and here. Instead, they may also bind sometimes. Say I'm just drawing a other one. They could bind here like this, and in that case they might break here and here. So whether a holiday junction will be broken in site 1 or site 2 will depend on how RUVC is bound. I hope you understand that. So it's a random chance that RUVC might bind in this way, or it might bind in this direction. Okay. So that is how mostly they also have kind of sequence specificity, and they prefer regions that have something like this. So if there is a sequence which has A or T and so 3 T's and G or C, that is a site that is most preferred for cleavage, and they cleave around somewhere around here. Okay. Yes, that is about E. coli reg-PCD pathway. So we need to talk about several things. We have to talk about Chi site and the significance of it. We have to then talk about reg-BCD proteins. To start with, yeah, reg-BCD are involved in processing the incoming DNA. And then we have to talk about, in this case, you have to talk about what do reg-BD do, what is reg-D, and so on. And then we have to talk about the rec-A processing of the DNA or the coating for strand exchange. 3' overhang should be exchanged, strand invasion should happen. And that is where we talk about rec-A protein. And then we will talk about branch migration, RUV-AB, and you have to also have function of RUV-A, recognition of holiday junction, RUV-B, that makes the hexameric ATPase motors that wind and unwind DNA, and as a couple they will result in branch migration. And RUVC is the nuclease that makes the cut. Whether it is cutting in site 1 or site 2 depends on how the RUVC dimer binds to the holiday junction. It has a modest sequence specificity. You don't need to by heart this, but you should know that it doesn't cleave everywhere. They have some kind of specificity. Now we will see about this. We will not discuss much because we have spoken about it more. It's about meiosis. We have spoken about exchange of DNA. This is about eukaryotes. In all of meiosis, it is a reductive cell division process in which diploid 2N cells will become haploid, or a diploid cell will give rise to haploid cells, right? And in that case, there is one more importance of meiosis is recombination. Okay, that is where, and it is mostly homologous recombination. We know that during meiosis chiasmata form, when first you have homologous chromosome pairing and then tetrad formation. This is something like tetrad formation. These crossovers are called as chiasmata or crossovers. And upon resolution, what you see is there is recombination. Again, what is recombination? Exchange of genetic information. So in one cell, in the chromosome blue one that we started, you have capital A, and both are chromatids, sister chromatids. So they would be identical. The homologous chromosome is non-identical. That means it is highly similar, but not identical. So you may have small a, small b, small a, small b. So crossover happens between non-sister chromatids. Okay, and there will be exchange of DNA, and eventually what you have after meiosis 1 is you have capital A, capital B, capital A and small b. That is a recombinant DNA. The other sister chromatid here is not recombinant. It remained the same way as it was in this case. Same way, you will see that this strand remains as it is here. Capital small a, small b, and here also you have small a and small b. Look at the other chromatid. We have small a. In this case, we should have had small a, small b, but what the result is small a and capital B. That is a recombinant. Here is one recombinant, and this is another recombinant. These two, the outer ones, are non-recombinants because there is no change in the genetic information. Yeah, I hope you remember these, right? These two are sister chromatids like this. These two are homologs, chromosomes we are talking about. And here interhomolog recombination have occurred between non-sister chromatids and so on. So that is how meiosis will generate, say, 4 haploid cells, out of which some of them are recombinants. And that is one of the main purposes of recombination. Meiosis recombination, people tend to say it as it is made to generate diversity. If you think in a very large scale at this species level, you can think of it as it is a homogenization process, right? So we will discuss what I just said now in the end of this course, and I hope we'll have a better picture by then, and that is when it's good time to discuss about it again. So there are several proteins that make meiosis possible, and out of which SPO11 is well understood. It is very much similar to that of topoisomerases. The activity of it, they make a double strand topoisomerase 2, they make a double strand break, and they are conjugate, they are covalently bound to the protein, to the DNA in that process. So SPO11 will, as a dimer, will bind to the DNA and make a nucleophilic attack, and it is bound to the products as such. And then when there are new partners, it will reform these bonds. That is, SPO11 is the one that is generating the double strand breaks. Okay, there are some sequence preferences are there where cleavage happen. Those are called as SPO11 cleavage sites. They are similar to or analogous to that of Chi sites we learned in E. coli, REC-BCD pathway. So the SPO11 is the one that is making the double strand break. Then those are recognized by MRX proteins, and they are processed. This strand here is processed to generate 3' overhangs. These 3' overhangs are now taken up by 2 proteins called as DMC1 and RAD51. They coat these proteins like this. I hope you can recollect that it is very much similar to that of our protein recase role. And these, because of that, they kind of exchange this strand or make the strand invasion possible, and then this is where polymerization will start. The same thing also happens on this side, and you will end up forming 2 holiday junctions, and then resolution will occur. Okay, the names change, the concept remains the same, right? So I will give a brief introduction on mating type switching in yeast. This is something very interesting, and I also want you to all think of it. What do you think is better? There is an organism, it just gives rise to 2 cells or 2 organisms. That is asexual reproduction. In other cases, it's like we give rise to 2 kind of thing. 2 individuals, male and female, and they give rise to offspring. And this is sexual reproduction. Which one is logical? Which one is better, and why? If you look at it, asexual reproduction is easy, simple, and it is cost effective. Once an organism dividing and giving rise to two, or no need to find a mate, to build a house, buy a car, get a job. Why should all these things happen, pay dowry and all those things? Why to go through all those headache just to have sexual reproduction? Right? Even if you talk about in animals, monkeys and so on, there is fierce fighting for reproduction, because it is sexual reproduction. The males have to fight a lot to get opportunities to mate. That means they have to invest a lot of energy, time, efforts, thought, and also may endanger their own status in their group, or may even be killed. It is not favorable at all. And sexual reproduction seems very nice. You don't have to bother anybody. You can reproduce by yourselves. But then if you see that sexual reproduction is there in many, many life forms, and why should something so difficult process, when there is a logically simpler method available, why should nature or evolution go for sexual reproduction? You know about a spider, a type species of spider. The female sits on the web, and the male is usually very, very small. So assume that this is the web here, the spider that is sitting there, it's huge, something like this, whereas the male is very tiny, like this. And this one, the male has to mate with the female to generate or to get reproductive opportunities. And when it starts mating, the female starts eating the male. I think the same is the case with mantis also. You have to look at how sexual reproduction, or in general anything about insects especially, they're amazing social structures. You can see ants, bees, hornets, wasps. They have several nice strategies. You should watch on YouTube or on National Geographic and other channels. You should try them more. The point is, why bother so much when there is an option, when something like this was possible? Right? Is it to generate? My question here is again the same question, which I would tell you will address it at the end of this course. Is it to increase diversity, or increase variation, or decrease variation? That is the question. You can also go and try to read about it. Is sexual reproduction increasing variation or decreasing variation? That is what I want. Is it increasing variation or is it decreasing variation? It should be either of those. Otherwise, there is no meaning of why sexual reproduction should happen. Often, people will tend to say that it is to increase variation. And there is also another way of thinking that's called the Muller ratchet. You can try to Google it up and find. It is rather to increase homogenization, to leave or to get rid of unnecessary or mutated genes from the population, not individual, from the population. We're talking about species here. Okay, so I'll just give you a brief introduction and I'll stop. Then we will discuss in the next class. Yeast, is a fungi. They are in single-celled fungi, and they reproduce by budding. Right? They just give out, and eventually they will divide. The smaller ones will start growing. That is asexual reproduction just by budding. But it also, people have noticed that there is sexual reproduction in yeast, and there are two types of sexes. One is called as A, and the other one is called as alpha, if that is right. Okay. And there is a way of a a cell becoming, I mean, sexual reproduction always happens between A and alpha. It does not happen between A and A. A is like one type of sex. For our ease, we will say, we'll call this as male. I don't think we should call it as male or female. We will leave it. This is sex type. This is A type, and alpha is the second sex type. And recombination always happens between A and alpha, right? And again, this is just another introduction. So alpha can, what people, when they were doing experiments, was they know that alpha, if there is an alpha cell, and it was undergoing budding, it is reproducing by budding, then what will happen? If this cell has given rise to a total of 100 cells, you would have had 101 or 100 alpha cells. Then is there a possibility for sexual reproduction? No, sexual reproduction does not happen. But what happens is, when they do like this, what they saw is, after formation of about 100 cells, some of them, um, say 76 are alpha type, and there are about, how many would that be? For 24 are A type. How did A type come in there? That is the question. So what happens is, alpha cell type gives rise to alpha cell type asexually, but somewhere some switching can happen. So one cell type, that is alpha, can switch to A type, right? And then it will keep giving A type cells. And again, somewhere down the line, A cell can give rise to alpha cells. And that is how a mixture that is thought to be of alpha eventually gives rise to a mixture of cells containing A type and alpha type. That means somewhere one of the cell, as shown here, can switch its mating type, and that phenomenon is called as mating type switching. It's a very interesting one. It will be a good idea if you can watch some animations that is there in the homologous recombination. It must be there. You can also try YouTube, and it's an interesting phenomenon to understand how mating type switching happens, and it is to do with homologous recombination. We will discuss that in the next class. Okay, please read the textbook. The textbook also has, this is whatever I'm discussing is from the textbook. So try to read, and we will know more in the next classes. Any questions?